Okay, ladies, so we're going to talk about complex inheritance, okay, which is different than your regular Mendelian inheritance, or dominance, as we generally call it. So, inheritance. Okay, so let's talk about it. We're going to discuss three different types of complex inheritance right now. The first is called incomplete dominance. And incomplete dominance is when the heterozygote, okay, so think about a heterozygote, the phenotypes are intermediate. Okay, now what does that mean? What does that produce? And generally, what we talk about is a type of blending. Okay, so blending. And let me give you an example that we often talk about. These are snapdragons. Snapdragons are the classic example of incomplete dominance. Snapdragons. Now, who knows what snapdragons look like? Well, I am going to show you. I love snapdragons. Snapdragons are awesome. And let's bring this down a little bit, see if we can do this. Alright, and if you remember, or if you've ever seen snapdragons, which are absolutely beautiful, I love them, if you press the sides of them, their flower opens up, and they are really beautiful flowers. So snapdragons, we're going to talk about this idea of blending. So here we go. Big R equals red, little r equals white, and so far nothing here has changed from what you're used to. However, when I cross a homozygous recessive with a homozygous dominant, what we find in our heterozygotes is a blending of the alleles. Okay, and you guessed it, the heterozygote is pink. Pink, woohoo, okay? And the reason for this is that the enzyme that is coded by that gene has varying catalytic ability depending on whether or not it's got it's a homozygous dominant, homozygous recessive, or a heterozygote in varying shades. So it's red to pink to white, and that is incomplete dominance because neither of those alleles are dominant over the other, and neither masks the other, and instead you get a blending of those traits. Okay? So the second example that we're going to talk about today, instead of incomplete dominance, and think about it as like incomplete. So if you think about dominance as, you know, if something is dominant, it is. It's the light switch is turned on, right? But incomplete means it's not quite there, right? Well, in this case, codominance, okay, is when actually both alleles are expressed. I often say that it's like being a co-chair of a committee. You have two co-chairs, right? Each one is in charge. Each is a chair. So they both take control over the situation. The same is true in codominance. Both alleles are expressed. Okay, so both alleles are expressed. Alright, so let's take a look at an example of this case. The example we're going to talk about here is sickle cell disease. And sickle cell disease involves the protein hemoglobin. And it turns out that the mutant form of hemoglobin, which is the sickle hemoglobin, turns these red blood cells into a funny shape. Okay, so these are red blood cells, and they turn them into shape kind of like a sickle. And if you think about your Russian history, you can think about the sickle, like the one on the communist flag. So let's take a look. I'm going to define my terms. Big H equals normal hemoglobin. Little h equals sickle-shaped hemoglobin. Okay, and I want you to recall what hemoglobin is used for in your body. It is used for transporting oxygen and CO2 from your body to your lungs to the air and vice versa. Okay, so that's the protein that we're talking about here. Okay, so let's go through each of the genotypes that are possible. So homozygous dominant, this is going to be normal hemoglobin, all normal. The heterozygote, though, contains two different alleles, both of which are expressed. So you're going to have some normal cells plus some sickle cells, okay? And the homozygous recessive is going to be all sickle. Okay. So hopefully that gives you some sort of clue or some sort of idea that codominance is really about both alleles being expressed, and that's different from incomplete dominance, where neither allele is dominant over the other. Okay, that's the big difference. Okay, codominance, think of like co-chairs, both are dominant. In incomplete, neither is dominant. Okay, so those are two classic examples of complex inheritance. The third type that we're going to talk about right now is sex-linked traits. And here, first, before we talk about sex-linked traits, we have to talk about sex determination. And this is going to seem probably fairly obvious to you, but I'm going to go through it and talk about some vocabulary words. So, if you remember, and as you well know, X and Y are our sex chromosomes. And these sex chromosomes carry genes for things that help us be male and female. And the X chromosome is much larger than the Y chromosome. The Y chromosome carries about maybe roughly 100 genes versus the X chromosome, which carries way more genes, and it's much larger. It's much more like a normal chromosome size, okay? And we're going to be doing a karyotype in class, so you'll see what's, you'll be able to compare those two chromosomes when we get there. Okay, so as you know as well, XX equals female, and that's the symbol for female. XY is male, and this is the symbol for male. Now, you have sex chromosomes, you have one pair of sex chromosomes, and you have 22 pairs of what we call autosomes, which are basically everything but the sex chromosomes. Okay? So just to clarify and make those distinctions, all of the genes and alleles that we've been talking about so far have been autosomal. Okay? But now we're going to be talking about sex-linked traits. Okay, so we need to talk about how we set up these Punnett squares then for sex-linked traits. And let me give you an example. We're always going to put the X and the Y on the top of the Punnett square. This is how geneticists do this. And you are geneticists. And we are going to put the X and the X on the left-hand side. Now, if I were to ask you, what is the % chance that any offspring that you have will be male, and what % chance will be female? You're going to say to me, Miss Wilson, that's a silly question. Of course, we know this. It's 50%. And I would say, absolutely right. How do you know that? And what would you say? Think about it. What would you say if I said, how are you sure that every fertilization event has the chance, a 50-50 chance of having a boy or a girl? Okay, thinking? Okay, I'm going to prove it to you. Ready? Watch this. X, 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 Y, X, Y, 50%, 50%. Fantastic. Remember that these represent the possible gametes of the parents, okay? And that means that this individual could give an X, this individual could give an X, and together they make a girl. This individual could give an X, this individual could give a Y instead, and instead we get a boy. But it's a 50-50 shot every fertilization event, okay? So let's now talk about this. What this sex-linked means, the word linked means carried on the same chromosome, okay? So carried on the same chromosome. Here we have sex-linked traits. Sex-linked traits. And sometimes they're also called X-linked, and that's probably more accurate in terms of terminology, because generally I'm never going to be giving you a question for Y-linked traits, because we don't really do that. There are only about 100 of them, so generally we're always going to be talking about X-linked, okay, and that's important for you to know. Now, since males only have one X chromosome, they will always show their allele that is located on the X chromosome. I'm going to say that again because it's really important. Since males only have one X chromosome, whatever is being shown on that X chromosome, whether it's a dominant allele or it's a recessive allele, that's the phenotype that will show up. Okay? So let's now do a classic example of red-green color blindness. Okay, color blindness, okay, and you do need to know this. This is a recessive X-linked trait. Okay, recessive though. So that means when we define our terms that big B equals normal and little b equals blind. Okay? Now, I'm going to give you an example here where we have our parents' generation. And the mother, okay, she is a carrier. And what does a carrier mean? It means she is heterozygous. Okay? Alright, now the dad, he is not colorblind. Okay. So let's look at what their genotypes are going to be then. Alright, so we've got, this is how we write this, big X, they're all big X's, but mom is going to be a carrier, so we put a superscript on the right-hand side of the letter. Since she is a carrier, she's got a big B and a little b. But since she's female, she's got two X's. Okay? And she is going to cross with her husband, who is not a carrier. So he's going to have just one little b and a y. Okay? Alright. I want you to see if you can set up. Okay, did you try it? Did you push pause and try to come up with your Punnett square? I hope you did. Here's what it should sort of look like. Let's do it. Now, I give myself a little bit of extra space in these squares. I make my Punnett square a little bit bigger because these X's and Y's can get really jumbly. So make sure you kind of give yourself some extra space so it's not all squished, okay? But this is what your Punnett square should look like at the end of doing this cross. Now, if I was going to ask you, what is the percent chance that any child, percent chance of any child being colorblind, you would say what percent? Any child. You would say 50%. Okay? If I said, what is the percent chance of a girl being colorblind, you would say 50%. Why? Well, out of all the girls, which there are two possibilities here, you have